Hey all, in this video on viruses, we're going to focus on bacteriophages, which are viruses that infect bacterial cells. And we're going to focus on their life cycle. Particularly, we're going to look at what we call the lytic and the lysogenic life cycles. Let's first talk about the structure of bacteriophage particles. These particles have a complex structure consisting of a capsid. Remember that a capsid is a protein structure that protects the genetic information or the genome of the virus. Bacteriophages also have what we call a sheath, and actually inside of the sheath here is a hollow protein tube that allows for that genetic information to pass from the capsid into the host cell. In addition, we have what we call tail fibers. These tail fibers function in the process of attachment. They will bind specifically to proteins or to other structures such as lipopolysaccharide on the cell surface that will allow for bacteriophages to attach and eventually allow their genetic information to gain entry into the host cell. Another important structure is this base plate, which we find on the bottom here. This base plate contains enzymes that will break down the cell wall of bacterial cells. This will help allow for the genome to gain entry into those host cells. Now, the lytic life cycle of a bacteriophage differs slightly from the lytic life cycle of eukaryotic viruses. In this particular example, what we're going to look at are 5 steps of the bacteriophage's lytic life cycle. In the first step, what's going to happen is that the bacteriophage is going to attach to the host. The tail fibers that are on the end of the bacteriophage will attach to the receptor, again, whether this be protein or lipopolysaccharide. The sheath is going to contract, and the base plate at the end of the bacteriophage is going to make contact with the cell wall. Now, the base plate contains on it lysozyme, which is an enzyme that we've talked about previously. This particular enzyme will hydrolyze the cell wall. And so what this is going to do is to prep the bacterial cell to accept the phage DNA. In the next step, penetration, that phage genome is going to enter into the host cell. Now, I should point out briefly that the majority of phages encode genomes that are made of DNA, and so that's why we're going to focus on DNA here. In the next step, and this can occur within a few minutes, we're going to see biosynthesis. The genome is going to be replicated either by a polymerase encoded by the phage or potentially the host DNA polymerase. The mRNA is going to be transcribed and proteins are going to be synthesized. In the next step, assembly, which is also referred to as maturation, the bacteriophages will actually begin to self-assemble. Proteins that are part of the viral particle are going to start to interact with each other. And what we're going to find is that that viral genome is going to be captured or packaged into the capsid by a process that's more complex than what we're going to talk about in this video. Finally, what we find is lysis of that host cell. The virus, in this case, actually encodes lysins, which are going to hydrolyze the cell wall, just like the lysozyme did at the beginning of this life cycle. However, these lysins are working from the inside out and are going to create multiple holes within the bacterial cell wall. This allows then for the viral particles to exit the host. So in total here, when we think about this in terms of time, this process can be as short as 30 minutes under optimal conditions. Actually, in E. coli, this process with some bacteriophages occurs as quickly as 20 minutes from the attachment to the lysis step. At the end of lysis, those newly formed viral particles can begin this life cycle all over again. Now, some bacteriophages are what we call temperate bacteriophages. They are ones who undergo the process of lysogeny. in which they integrate their genome into the host genome as shown in this picture. Upon integration into the host genome, this viral genome will remain stably in the host genome under optimal conditions. However, in times of stress, what we find is that the bacteriophage genome will be excised from the host genome, and the bacteriophage at this point will undergo biosynthesis, maturation, assembly, and ultimately exit to leave the host cell. Now, an alternative mechanism occurs when bacteria are not under stress. Bacterial cells that contain this integrated viral genome but are not under stress will continue to replicate, and as they do, the viral genome will be replicated along with it. You could imagine that one benefit of this mechanism is it allows for the virus to be propagated or passed along to multiple generations of bacterial cells. If these populations of cells then become spread throughout a region or an area, that bacteriophage ensures that its genome and its genetic information will survive and be passed along to future generations in that disseminated area. I want to finish off by just briefly comparing this lytic and lysogenic life cycle. In lytic life cycle, we find high levels of replication very quickly, maybe 20 to 30 minutes from the point of attachment to the final lysis of that bacterial host cell. And this process is incredibly productive, potentially producing on the order of 50 to 100 bacteriophages per bacterial host cell. In temperate phages, during the process of lysogeny, we find a different mechanism here. It seems like these temperate phage are playing a long game. They replicate only in stressful conditions in which a cell may be stressed and may be on the verge of dying. Otherwise, temperate phages may retain their genome in their host cells indefinitely. And this can be quite beneficial. If the goal of organisms, living and in this case non-living viruses, is to pass genetic information on from one generation to the next, then lysogeny can be incredibly beneficial in that it relies on the host cell to perform the replication for it.